I'm not where I want to be. That stupid mouse, I ain't touching it. It's like a bug. And to click it, you had to click it as you go by. Bam! And hope you get it. But actually, I got to right click it so I can say open. I got to tell them about that. And I can cross it.
When will we have? Well, we'll do more lessons before we have a paper test on the other.
Yeah, we'll probably do a few more lessons first. I like kind of having all kind of a lot of stuff. You're going the lessons I'm doing is what they said. Be sure and teach this. Be sure and teach this. So it's not what I'm pulling out of the sky. I have a list of things they want me to cover, and sometimes it'll say cover the cell membrane, and it'll say over here do osmosis of the membrane. So I'm doing the same thing twice. So this is about DNA, RNA, and protein synthesis, and you've already had a good dose already.
Yeah.
I mean, so this should be a really easy lesson. And this is another picture. I like this one here, though. This shows the big unit, the small unit. It shows the messenger going through it. It shows the three sites, the arriving site, the protein site, and the exit site. It shows the weird-looking tRNA with the amino acid on top of it. It shows this thing landing. And what it doesn't show you is the actual letters, because you know, doggone thing will be still. If that was U, then that's got to be A, you know that. So it don't show you. I might one day go back and put them on myself. But this will be the anticodon of what's down here. And he'll slide into here. And then this will be handed off to this. And then they'll go across together. And actually, 1, 2, 3, 4, 5, 6, 7, 8, 9. There's not been 10 before going through this thing, dropping off the acids. And somewhere, you know, there's going to be a termination site which has a codon that nothing codes for, and that's the message we're done. And they're going to drop it off. That's pretty cool.
Hey,
Right.
I hate to stop you for a second.
Alright. So anyway, oh, that's wonderful. I was about to make a fool when I called the folks. They come fix your computer. Fix that. I didn't realize that. I'm glad you saw it. Alright. I'll know next time. Look at that. But anyway, I like my start. I always like to have a starter picture of my first slide to give me an idea where I'm going with this thing. And obviously, this is a 5N, and that's a 3N because it's going this way. This has to be 3 and that'd be 5. And you know, the 5 has a point, right? So the 5 is always coming in like this, and so that's how I know that this thing's traveling in this direction. Also, the A is the entering site, and the exit, it's got to enter this way and leave that way. So it comes in loaded, it leaves empty, and it passes the acid on to the next chain. And all these are just amino acids, and that whole thing, I'll call that a protein. Okay.
You said the E is always where 5 is, right? So this chain is coming from the right side going to the left.
That is correct. It's traveling this way.
Okay. So if it was flipped, then the A would be where the E is, like it would. Okay.
Right. And then it'd be going the other direction. Exactly.
Right.
Yeah. A leaves. Because when you're going to plug this thing in, you plug it in from the A side, it goes from the A to the P to the E and gone. Okay. Let's look at the first one. The whole question is about the relationship between these two. And the first time I saw it was back in 1902, even before we discovered DNA. We knew things were going on, we just didn't know how it happened. A lot of times we know things are happening, but can't figure out why. So Garrod was the one who actually was studying a thing called alkaptonuria, which is the problem with the urine, and there's a buildup of a chemical in the urine that turns the urine black. And that's how he began thinking. And if you have this condition, your joints ache. And often that you'll be so bad, but by the time you're 50, you're probably having knee replacements, things like that. This guy was a biochemist. So he had a background in chemistry. So he knew there was a pathway that, you know, this chemical happens, and then that chemical makes this happen, and this chemical makes this chemical happen. And if you remove any step, then something breaks down. Well, he was wondering about that, and he had a hypothesis that he put together that what caused the black urine. And this is an acid called phenylalanine. And if you have too much of this, you get a problem. Now, there's a 4-step process to get rid of it. It's a 4-step pathway to get rid of it. Now, when it's over, and I can't even say that word, maleylacetoacetate, something like that. When you're done, this is what you have, and this stuff won't bother you. But if something happens to not get to here, and say this is going to be produced on the way to making this. So when this is made, this should be gone. I use this to make that, right? So if you find this present, then you know that something's going on, because why is he here? I should be seeing this. Because this is an intermediate step to get to this, and this is my problem. So the guy, he thought there might be an enzyme responsible, and the enzyme, he called it. Now there's the word I hate these words. Homogentisate or genesate. And the hormone that breaks it down is just oxidase. The same word in oxidase. And if you don't have this, then you can't make this become that. And if you have this, you're good to go. This, you're in trouble. So during the 4 steps to get to here, this is one that, and all of us here, we won't find this because we're breaking it on down. We're good. Our urine's not black. We don't have joint problems. But the guy began to wonder what would make this stay there. And he thought, there must be an enzyme that's not there. Because if you had the enzyme, you'd lose this. So this is the enzyme that's probably a problem. If you don't have this, you can't get rid of that. If you can't get rid of that, you won't get here where you want to be. That makes sense so far? It's a chain reaction pathway. So this guy, he's just thinking out loud. He's just thinking. Okay? Oh, here's homework. This blew me away.
That's all right.
You may have heard of aspartame.
Yeah. It's sweetener.
And there are a lot of sweeteners. There's equal, sweet, low, Splenda. And there's a connection between this amino acid and that sweetener that causes people to have problems using too much of a certain sweetener. And I'm thinking, man, I want to use sugar instead.
Yeah.
Than the sweetener.
Use an organic.
Exactly. Go ahead and so the question is, what's the connection between these two? And you might Google just that very question and get answers. So I want to know, first off, there are 2 sweeteners, and they really are this, but they're not called that because books wouldn't buy them. They have the name that you recognize. I want to know what those 2 sweeteners are. That's actually aspartame. And then, remember now, this is what you want to get rid of. Now, there's a connection between this stuff, which is artificial sweetener, and that you want to get rid of.
What's
All right?
in it?
Huh?
It's in it.
And then, if you have too much of this stuff, how does that affect your health? Like, Diet Cola, you think it's so good for you? I want to think twice.
Diet or something?
because we use another kind of sweetener instead of sugar. So you are not getting the sugar, but you're getting something else, which might be worse than the sugar.
So pretty much that's just like medicine.
What's medicine?
I'm just saying this is similar to medicine. Like they say you take medicine, but you have 5 side effects from it, so you have to take something else.
That's true.
to counteract
Yeah,
that.
I know what you mean now. Yeah. And next time I meet, be next Wednesday. That's why I'm kind of waiting. I'm glad that we did it today and give y'all a whole time to get ready.
Almost.
And you can write it on paper and give me the paper, or you can email it to me. I don't know how you get it to me. Now, I'm going to basically read your answers. Now, if you email it to me, I can't make comments on it and let you know what was good or what was bad about it. But if you give me paper, then I can make comments to you. Because when I was doing my research, when I was making the PowerPoint, I came across this. I went, I didn't know that. Oh God, I got to tell the class about this, and I made homework out of it. So, and I did one that I didn't do y'all because I'm still progressing. And the other class y'all took, we did one on diesel fuel that you didn't see. There's 3 colors diesel fuel. There's brown, red, and blue. Did y'all know that? Yeah, there's 3 colors. Brown's the one you buy in the gas station. That's the most expensive of them. We process it to make it burn cleaner. Now, if you're a government vehicle, or if you're a Red Cross vehicle, or if you're type of a non-profit, you can buy the red color diesel fuel, which is cheaper. Off-road. And if you're a farmer on your John Deere tractor, you can burn the blue kind, which is really cheap. But Lord, if you get caught in your car with that blue fuel, that red fuel, it's thousands of dollars fine.
10,000.
It's terrible.
What?
For a gallon of wrong, they'll burn you up.
10 grand is what the fine is.
Yeah. So farmers can save. Now, if I want to drive my John Deere tractor all day down my fields, I want to use the cheapest I can find, right? So and it's dyed blue. It's blue.
Oh, it's dyed blue.
It's dyed blue.
Okay.
And it's dyed blue, and they can use that. Now, if your pickup truck, which runs out of diesel fuel, and you siphon some of your tractor in that truck, you better hope the cop don't catch you as you go down the highway, because they'll take that. They have a siphon, and they'll pull. I know the big truckers always have it happen to them, because they must. The over-road truckers haul from here to California. They have to buy the expensive diesel fuel. They would love to buy the red stuff. Now, you know the big trailers with the big refrigeration units that haul like meat inside it, and you hear the thing always running. That diesel fuel can be red. That burns that runs the refrigerator. So you may have red in the big fuel tank on your trailer, but if you take on your truck but not be red, you can lose all your profit. And if you're a trucker, probably lose your license too. So I never knew that kind of stuff. I got a friend who's a farmer, and I knew he had that kind of diesel fuel. And see, that Sudan stain we were using, we use Sudan and tested. That's the stain they use to stain the red diesel fuel. And I was doing some more research. I'm always making my labs better. And I go, I didn't know this. And so I worked a whole other thing about within the lab for the Sudan that this is the same stuff that they use in diesel fuel. And in England, it's called low tax red, because in England the home heating oil can be red diesel fuel. Low tax red, it costs less.
And it's the same stuff.
It's the same stuff.
That seems kind of tyrannical, though. I mean,
I know it does.
Well, that's your government for you.
Well, that all came about back in 1994, where they passed a law to clean the air up. So, well, if I own a diesel car, I got to gas up at Spectrum, and that diesel fuel is the top dollar. It's the most refined. But if I have a farm and I got a John Deere tractor on my farm, now here, first thing, I don't know why I'd go to buy the fuel, first off. But where I go to buy the red, I know farmers know that. I know they can't sit out here because you might put in your car by mistake and you'd be in trouble. So I think what happens, I think I have my own in-ground fuel tank. And I call, hey, Mobile, would you bring me some diesel fuel, red for my tractor, for my farm?
They sell it at the gas stations. There's a gas station in Waverly Hall that I used to.
How do you know which is which?
Because it says off-road diesel on it.
And that's the red?
Mhm.
So you don't put that in your truck, though, do you?
I mean, I have, but
Yeah, you hope, and
it was a while back.
if they stop you for a license check, they might not check your fuel.
Right, but when they really look for that kind of stuff is if you're like, have DOT numbers for
Yeah.
transportation, or if it's
You may be straight then.
commercial vehicle,
Right, right.
you know, or something like that. They can stick a dipstick in there, and
And you see it dripping.
it'll stain the thing or something
Our
like
tree
that.
service, we have 3 diesel chippers.
Mhm.
All our other trucks are gasolines. We don't even fool with the red. We just go in, once a few gallons of diesel. And we check our level by we have an old broomstick handle, stick in there. You got a foot, that run a day, you know, and you see the brown dripping off of it.
Yeah.
So I guess if you had red, you see the red dripping off now. We'd be legal in that chipper.
Mhm.
But we had us a diesel truck. We better not be caught with that stuff in that truck.
But, I mean,
And to save a few nickels a gallon, and for $10,000 fine,
Oh yeah.
it ain't worth it.
They'll
Yeah, that would
but
eat
will
your
it
dinner.
mess up the equipment?
Huh?
It won't mess up the equipment. So why they want to charge you all that money then?
Because they want to make more money.
They want to make money. As a matter of fact, when I did a reading on why they did it, there's 2 reasons. They wanted to make the air cleaner. They wanted revenue for road construction. So when you buy that high dollar, you pay them for the roads.
Hey, that's $1 now. It used to be a lot.
It used to be cheaper than gasoline, and you had a reason to buy it then.
Now it's more than premium.
And I don't know why it is. And so now the folks that bought the diesel cars to get ahead financially are now behind.
My truck's diesel.
You still run diesel in your truck?
I have to.
Do you find it very, very filthy? Do you hand smell when you use it?
Yeah, when you pump, when you.
Do you wear gloves when you pump?
No.
Some folks wear gloves.
Oh,
'cause
yeah,
it stinks. It stinks.
Right. It's kind of hard to get off your hands when you wash
Yeah,
it,
it
but
stinks.
no, I do, and I even make some of my own alternative fuels for it too.
Why now? Also,
Is it legal to
there's
do that if you have like your own business and if you made your own fuel? Is that legal to use your own?
like making your own liquor. That's against
They
the law.
don't say no,
Is
but
it?
they.
If you pay tax, you can make your own liquor. See, the bootleggers, they got hit by the revenuers because they're not paying tax on the moonshine. If I make moonshine and sell you a gallon for $5 here, I didn't pay tax on that. That's why it's illegal. Making moonshine is legal if I pay tax. Bud does it, Schlitz, Miller does it, because they pay taxes. But a guy in his backyard running off a still, brewing that stuff that sells his neighbors for like, a couple bucks or half a gallon.
Is it only if you're running a business off of the fuel that you're using?
Don't worry about business. A lot of folks play with the fuel. And a diesel motor is more forgiving than gasoline.
Oh yeah, I run.
So he can, I wouldn't even do it. No, I wouldn't even consider my gasoline. But gasoline's real finicky. That
Yeah.
diesel, they can run all kinds of stuff in diesel.
I run waste motor oil with gasoline mixed together.
Oh yeah,
I run all kinds of stuff in it.
And it won't hurt it. A diesel is it's a very heavy, tough engine. That's why the diesel is used for a long time. Is all the big tractors are diesel because these big huge tractors, they're like 40 horsepower. But the way they're geared, them 40 horses go a long way. You got a car out there for 300 horsepower. You don't need the horse if you got it geared right. I mean, when I drove a dump truck, I drove it. My father bought 2 dump trucks, and we drove McMath. One of the dump trucks was diesel powered, one was gas. I drove the gasoline. I could blow him away. As far as takeoff, underload, he couldn't keep with me. But my truck would run hot sometimes. I had to stop and let it wait a little while. He'd come plugging on by me. And diesel had power though. Because one time he backed into a pit to get loaded. And we got loaded, he gave too much gas, and the draft had just twisted right off. Flop, 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 flop. And mine never did that. You burn the clutch up if you do that. And the parts we found out, if my gasoline gives out a gas, I just put gas in the tank and
You better not do that.
when your diesel gets out of gas, you get air in there. Oh boy, you got to bleed the lines. So we don't have any more gas. Our tree, we have like 15 trucks we use for our tree service. They're all gasoline powered. And gas now, we haven't charged more for a tree job because it costs more to run the truck. So we pass on to customers. When the gas goes up, we're going to accept the tree job just cost $100 and that costs $115. You know, everything passed on to y'all. It's not like everywhere though. You gotta make a living. We had to cut back on workers because we couldn't afford to pay everybody, so we're down to a very small crew. We make our living and we buy our bread, and a lot of our trucks don't get run, and we have a spare truck will sit right there and fall apart. Tires will go flat on you. You got to run it to make it work. It's the idea of I'm going to buy 3 trucks and run one of them, have 2 for standby. That will not work. When the one you're running won't run, the one for standby, his battery's dead. Oh, crap! So the idea of having a big fleet for standby, we now know does not make any sense at all. Because you got to run it to maintain it, because then you know something's wrong with it. So we learned that lesson. We sold a lot of trucks off that we didn't need anymore. So there's your homework, and you know, it's going to probably open your eyes up too to what, because I went, oh my God. So I've changed my eating, what I use in my coffee now.
See, I love Diet Coke. I like it better than regular Coke, so I don't even know if I want to know how bad it is.
Well, that's what, you might not want to know. Anyway, you're going to have to get it homework then.
Make it learn.
Well, no. Do you recall when it said saccharin causes cancer?
Yeah.
And they had to come back and they said, yes, saccharin causes cancer if you eat 1,000 pills a day. Now, who's 1,000 pills a day? I mean, the fact is, you can say saccharin causes cancer and shut up. But that's not, but how? I don't use 1,000 pills in a day. I use maybe 10 tablets to make a gallon of tea. And that gallon lasts for days and days and days. So I'm not using enough to say, oh yeah, it causes cancer. If I take the whole bottle, no, I don't have to drink it down. But you know, sometimes people will tell you things like that and not tell you the other side of the story. That, oh yeah, well you got to mainline it to do that. Otherwise, all right. This is the. Let me get back where I was. You remember the pathway I said you want to get to here? That the M word, the Mali word. It looks awful, but that's what you want. If you stop too soon, you get the black urine, you get the joint problems. So here it is. This is where you start. You want to get rid of this. This is where you end, and you're good to go because this is good stuff. Somewhere in here, this never happens because this has a problem. And this stays in your system, and this is what turns the urine black and tears your joints up and that kind of stuff. So that's what Gerard was wondering, could there be an enzyme that maybe is not working? Because we often wonder what the key was between DNA, enzyme, and proteins. Okay? Well now, you hear Mendel? Gregor Mendel, the father of genetics, and he used peas. And peas, some grew tall, some grew short, and some had red flowers, some had pink flowers, and some had yellow seeds, some had green seeds. And there was always either or. There's never a pink flower. When you cross a white flower and a red flower, you either got white or red. You never got a mixture of them. That's a Mendel trait. If you crossed a red rose, bright red, with a startling white rose and got a pink rose, that's not Mendel. Because Mendel says you're going to get one or the other one. Because one's going to match the other one. They ain't going to blend. So anytime you hear Mendel, our ears are Mendelian. These are traits. Some ears come right down into your head, and some hang down like mine do. Some flop down. Well, that's the Mendelian trait. And the floppers are dominant. And then the ones that go straight in are recessives. It don't mean a hill of beans, that's just the way it is. Some folks can do this, and some folks can't. I play harmonica because I can do that. And I know at Hardaway, I was in my room one day, and I was playing Dixie. And a lot of folks don't like Dixie, as black folks say. Do you know Dixie was written by a black lady? She wrote that song in jest. Oh, I wish I was. No, she didn't either. You know what I'm saying? She wrote the whole song as a joke, but the tune caught on. And in the Civil War, when we were fighting up north, they did wish we were back in Dixie where their families were. So it took on a whole new thing. But I was in my class one day, and I was playing like Amazing Grace and Precious Memories, and somehow I went from there, and I was playing Dixie before I realized it. Because Dixie is hard to play on harmonica, especially the part that goes back and forth. And in walked the black guy. I went, oh my God, I'm in trouble. He goes, what's that song? It's so pretty. I went, oh, just something I made up. I didn't tell him what it was. And my thought is, how many folks don't like Dixie because of some brain type have never heard the song at all? And when you just play it without the words, it's just the prettiest melody. Well, even the words, though, if you listen to the words, I can see a black lady writing that as a joke. I can see a rebel soldier, far from home, in combat, wishing back where I can see that too. So it goes both ways, but it was written, I understand, by a slave. And that's just, folks didn't know that. And some folks don't like that idea. Can't be, no way, I'm not Dixie. Yeah. Some folks still live in the past. And they'll never get out of it. It's unfortunate. And you know, education solves that problem. It ain't educated, it's the ones that are not educated who have those stereotypes. They've never taught, learned anything different. It's sad but true. So, if you, the phenotype, now here's phenotype. You see my ears hanging down? That's a phenotype. My genes that make that is my genotype. I can roll my tongue. The rolling is the phenotype. The genes make it possible. Oh, I was playing Dixie, and the guy who was talking said, how long you played that mouth harp? It's a harmonica, mouth harp. I said, oh, well, I remember. He says, can you teach me how? I'm like, you got a harp? He said, no, if I can buy one, what kind should I buy? I said, well, get a C harp. C, everybody can play C. But so I have a harp every chord. C harp, A harp, D harp, A flat, B flat. You can't change chords on the harp. You got to get the harp. I got a whole case full of harps, like in church. I'll say, what key? E. I get that harp. And you can play cross harp too if you're good at it. I said, well, let's get one of them. I said, oh, before you start, can you do this? He goes, I said, you can't roll your tongue. I said, it's just, he couldn't roll his tongue. I said, I can't teach you how to play a harp then, because the little holes are so close together. You roll your tongue to make air go on one. Otherwise, you hit 3 holes at one time, which is nice sometimes. But you play a harp, you want one hole to blow. You draw and you suck. And
I didn't know you had to do that.
What?
With the harmonica.
What about it?
I didn't know you had to do that with your
What's the
tongue.
tongue?
I thought you just.
I don't anymore.
I have a question.
I don't anymore. You can also put your tongue flat like this and blow in one hole. But I do it now because I can play harmonica. If I couldn't do that when I first started. I had to do it this way. And it worked great. And it's just a matter. And this one, you know, in my car, this one I got in my car. I hear the radio, turn it right off, now I'll play the song. See if I can play it. And I play by ear. Yes.
The same way that we like the fake sugar, like the aspartame or whatever, do bacteria, anaerobes and aerobic bacteria, yeast, do they eat that too?
If you say aerobic, anaerobic, what would you say?
Both of them, because you need both. But do they eat
Yeah,
that too?
let's break it down. I want to do a PowerPoint, and I don't know why, but I know honey
That's man-made, right?
Well,
That's
honey
man-made.
won't spoil. I wonder why it won't. Honey won't spoil.
It's antibacterial too.
I don't know why. What is it about honey? Because why couldn't you use that and put in food and solve your food spoilage problem? Now, am I wrong? Honey won't spoil, right? I've heard that. Honey will not spoil.
It won't.
And I'm wondering why I want it, and if I find out why, can I not put that in like mayonnaise, and so it wouldn't spoil either. You know, we refrigerate food to stop spoiling right come back here. But I've always heard, and I never did research, and every time I think about it, I forget about it, but I want to know why it won't spoil. I want to know what is about it, and then could I take that to another level and use that to maybe solve the food spoilage problem in the world? Because so there's something about honey, and I don't know what it is now. When you have honey, would you call it sweet? I don't call it sweet.
Oh.
I sure if I call it honey, to me is not sweet. It's pleasant, but in my mind, I don't. And some people don't like honey. Some people don't like it. But when I've had honey, like I like to dip my chicken nuggets in honey. That's good to me. But it's not like. But I wouldn't want to do it in syrup though.
Biscuits.
Well, I like syrup biscuits. I like syrup, butter, and syrup. Make it real fat. Anyway, here's the point though. This enzyme is controlled in Mendelian. If you get any dominant gene, it'll be all right. If you got recessive genes, you got a problem. And I'll tell you about more Mendelian later on.
My question is, do they produce energy the same way with fake sugar as they do with real sugar?
Yeah, you break it. How you do break the bonds? Just break the bonds. Just because it's organic versus synthetic, I mean, a lot. I mean, when you burn gasoline, you break the bonds, and it gets hot. Your car is hot, and your motor is hot. But at the same time, your pistons are being driven. Of course, how often you change spark plugs in your diesel motor?
Never, because there are none.
You had no spark plugs. Would that be maintenance good? Because my truck, I got a spark plug. The truck's skipping. My gasoline has 8 spark plugs in it, and they got a spark. He has a hot plate. They're totally different engines, and his can be much more abused. Well, mine, you foul them plugs, it won't crank. He got no plugs to foul. His will crank on urine sometimes, I think. Okay.
And they run a lot more compression.
Oh yeah.
Way more, like
Yeah.
800 and something pounds per second.
When you stop your car in the hill, put it. Sometimes the engine that you roll with it,
Yeah.
diesel won't do it.
No.
You put a sucker in a gear with a diesel like a stick shift, you ain't gonna spin that motor over. It's gonna sit there until Hades freezes over. Here's what Jared said about this enzyme. He says, first off, he thinks the gene's the problem. He thinks that the gene is the problem, and the bad gene makes the bad enzyme. Well, that's the first time anyone linked genetics to making proteins. Before that, no one ever thought that genetics had anything to do with proteins. They thought it was strictly something being made. See, they always thought the nucleus made your proteins. I don't know how they got that when prokaryote has no nucleus. I mean, that should have been seen through right there. But he was the first one to ever think that, there might be something about genes, might be making these proteins. Now, genes are DNA, right? Now, we know now DNA with the genes controls every protein you make. And we know proteins, some are enzymes, some are hormones. Now, we know the nucleus contains your genetic material. We know that for a fact. And we knew that DNA was the material. We didn't know what it looked like though. So we knew DNA existed for a long time. We just didn't know what it looked like. Now we discovered the look in 1952. We knew about DNA for a long time. We didn't discover DNA in 52. We discovered the double helix. We've known about DNA for a long time. We knew it had something to do with genes. Even Gregor Mendel, he was doing his research without knowing that it was all talking about genes. So don't think that we discovered DNA in 1952. We didn't know. No. We discovered what it looks like. We knew about DNA for a long, long, long time. We just didn't know what it looked like, and that's when Watson and Crick and Rosalind Franklin came in. So they thought the nucleus was where everything was assembled, because it's ribosome. It's so far from the nucleus, it ain't funny. But back then, they had just used what they made sense to them. They didn't have the electron scope. They didn't have all the research we knew. Didn't have Watson and Crick yet to tell them how things really were. But this guy, he was on the right track. He was the first one to ever say, you know, I think those genes and those proteins, there might be a connection right there. No one ever said it before him. Well, there's something I'm going to tell you about that it is probably, and you might know, I do, it's one of the most famous experiments ever was conducted that clearly showed the nucleus does not make proteins. Because if you remove the nucleus and it made proteins, then would you think protein production would stop? You would think it would stop. Well, they actually did that, and the protein production kept going. I mean, because if my motor makes my truck run, we think, and I remove my motor, and it still runs, we go, oh my God, it ain't the motor. Well, we thought the nucleus made the proteins, so we took out the nucleus and still made proteins. We go, oh my God, it ain't the nucleus. We began wondering, well, what is it then? And we kept doing research. So, acidobularia is a type of a plant, and it's the coolest thing because it's got a foot, and in that foot you can find the nucleus. If you cut the foot off, you won't kill the plant, but you will remove the nucleus. So here's what we're talking about. That's a look of it. It's a little teeny thing, it looks like an umbrella on top of it, and right here's the foot. And that's what the nucleus is. Now, this thing is called the rhizoid. And because the nucleus is down here, if I remove the nucleus, then new proteins should not be appearing, because if the nucleus makes the proteins and I remove the nucleus, then no new ones should appear. So because the nucleus is down here, it was easy to just lock that sucker off, cut it off, remove the nucleus, and see what happens. And this is a life cycle right here, where this part makes those spores, and they hit the ground, and the spore case develops. Now it comes into the seeds, and the seed makes a prothallum, and the prothallum grows up, and the nucleus stays behind. As it grows up, the nucleus stays behind. It grows up, makes another umbrella, and the cycle starts. And these are little bitty tiny plants.
So they're single celled?
Yeah. This thing is teeny. And we can remove this part of it. And then nothing should happen up here, because that's where the nucleus is. So here I thought, if the nucleus is making the proteins, and I remove that rhizoid with the nucleus, then don't you think protein production would stop? We thought that. So to enucleate means to remove. So problem was though, they kept living. They kept making the proteins to keep them alive for months. And the protein synthesis, it went on for 2 more weeks after we removed the nucleus. So we're going, ain't the nucleus then? If proteins still be produced, and this thing can live that long, it ain't the nucleus making the proteins, it's something else making the proteins. So that was the keynote research that showed us that the nucleus is important, it's not making proteins. So what the Spirit did was that cell could make new protein even without the nucleus. So obviously the nucleus was not making the proteins. Now another study came along, suggested the ribosome. So we first had to be told, see, if we kept thinking nucleus, would we even look for ribosome? There'd be no reason to, would there? So when we realized that it ain't the nucleus making protein, we said, then what could it be? Then we started looking again, and we found the ribosome and go, that's it, that's the one doing it. Now, that showed us why that little cell would not die if I remove the nucleus. He will die eventually because who has the instructions? The nucleus. You know, the messenger that's out there, he's still out there. When I remove the nucleus, he's still out there doing his job, right? And who made him? Now, when he finally gets all wore out, and no one knew it, then it'll start to die, right? But when the nucleus is removed, we remove the capacity to make more messenger, but the messenger that done been made is still doing its job. And that's why the thing did not die, because the ribosomes making the protein still had the instructions, even though the boss is gone. That makes sense? We know that now because it's 2012. But back then, they're going, what in the world is going on here? I ain't sure I want to live back then. I like knowing what I know. Of course, in 100 more years, they might think we were backwards. How could they believe that back in 2012? Idiot morons. And, you know, that's it. Time marches on. All right. So now we know it ain't the nucleus. And so now we know it's the ribosomes. They're the ones making the nucleus. And how we figure it out? The electron microscope. We can finally see the little suckers. Without that electron scope, we might have, I don't know. You can't see them, you don't know they're there. But that scope really made, it made so much things. You know, the ER and the Golgi apparatus that we now can see. That scope opened a new world to us. I don't mean the scope we have in the room there. I don't mean that kind of scope. I mean the electron scope. That thing goes up maybe 1,000 times. Electrons go up 200,000 times. We can see inside something that we never seen before. So, only problem with electron scope, it's got to be dead. Nothing alive. First off, you can't put a leg in that sucker. You got to put the part of a cell to see what the part is. So we're not looking. Those scopes might see, well, they can't see a lot of cells. They can see the cell, they can see the nucleus. They can't see all the fine grained stuff, but electron scope can. I often what electron scope costs. I don't know what they cost. Probably more than my salary is though. I wonder if we sold all our other scope we have, could we still afford to buy electron?
Look it up, see how much they cost. I need to know that.
Oh, I got pictures of my new scope. I'll show you my link. This is electron scope.
An old one.
An old one, you better believe it. You know about Palade, right? And this is a guy who was given credit for discovering the ribosome. This is a guy who let this guy borrow the electron scope. He won a Nobel Prize, but so did this guy. And this man is like me showing Katrina the scope. You would discover it, but I'd be wanting to show you how to let you borrow my scope. All right. Now, this guy, Claude, he discovered the ER and mitochondria. I mean, we're talking about pioneers of microbiology. And I wasn't born yet. I mean, I'm born in 50. In 46, Claude had this guy come to New York. And there he showed him his first ever electron. The guy goes, oh man, I gotta get one of those. And then in 55, the bumps, see, this guy had then discovered the ER. So his underling said, watch those bumps. Watch those bumps on the ER. Well, then he said, then those bumps, those are, he discovered these first and the free floaters next. See, he didn't know about these because he wasn't looking for it. But when he was looking at the ER along with Claude, he goes, what's those bumps? And then Palade goes, I'm going to find out what they are. And then Palade was trying to figure out those bumps are ribosomes. And he kept looking, hey man, they're floating around the cytoplasm too. So I find ribosome in two places, hooked to the ER, or just floating around. This guy also found that the stuff in the ribosome was ribonucleoprotein, RNA. That's why he won the Nobel Prize. He discovered a whole lot of stuff. Then the two subunits, he discovered that with the electron scope. He won the Nobel Prize many years later. But the problem was, though, Palade didn't name it ribosome. Roberts was the one who said, that is a ribosome.
How come you don't hear anything about that woman?
About Roberts?
The woman that I was telling you about, the Peterman lady? When she
Don't know
was...
it. Don't know it.
I mean, you hear about some of it.
If you don't write things up, and I suspect she didn't publish properly.
Well, there was a bunch of places that I found it in, but it was, you don't hear that much about her.
But you don't, because see,
Side by side.
if I were to find the ribosome in 1940, but I don't write it up, but I told you I did, and you might tell the world, but if I don't write it up, will I get credit for it? I suspect she didn't publish. That's how you find out. You publish, and people will discover your work.
Yeah, in some of the books that I saw, he actually gave her credit, George Palade, for being the mother of particles. Because
True, but the particle...
She was the first one to isolate the animals.
Isolating it and naming it different things of it.
Well, I mean, actually...
Because she isolated it and used her name for it is fine. But the name that we all use now is not Peterman, it is ribosome.
Right.
So apparently her name didn't stick for some reason.
But I mean, they were researching and finding this...
I agree with you. I don't know why.
And her name is not mentioned.
I don't know why. But I suspect probably because she didn't put the word out. If I'm working here with you four people, you know what I'm doing. I don't tell anybody else who will know I did it. Now you might write a book. You might become famous and say, "Oh, Mr. Tell, he told me this stuff," and that's how we find out about her. Somebody else said it. Mm-hmm. So I don't. It's not any type of a shun on her part. It's just that I don't think she really published it. But this guy is giving credit for the word ribosome. I don't know if she used the word ribosome. She used Peterman particle.
Yeah, well, it wasn't named yet.
But the problem is, though, that Peterman particle now is called the ribosome because he said ribosome. He didn't. I agree with you. He wasn't the first one to ever see it. She did, but she didn't name it, so she don't get credit for the name. But this guy here is the one giving credit for seeing it first, and I think she came before he did. She was before Palade. So that's that's stuff in novels. But I really suspect back that day and time, you know the saying, publish or perish? Mm-hmm. Professors back then, if they want to keep their jobs, they had to do research and publish. You know, the two guys who discovered DNA, Watson and Crick, it was a race. They were racing to discover it. Now they had Rosalind Franklin and Maurice Wilkins also in the race against them. Mm-hmm. And then two of the guys, father and son was in the race. There were three teams racing to find the DNA secret. There was a race. And apparently Watson and Crick. And the trick was, we got it. And Rosalind Franklin and Maurice Roberts, they published in the same magazine Crick did, but Crick article was on page 20 and theirs on page 50. He got credit. Same magazine. As you read a magazine, who you read first? Watson and Crick. Same magazine published it. But Watson and Crick got the credit, got the Nobel Prize, everything else for it. It's just that stupid. It's just that stupid. I mean, and now, I tell you right now, they didn't share much stuff. I don't want, you know, if Roger and myself are competing for the same discovery, and I discover something that's going to help me beat him, I can tell him about it. Then he might beat me. And see what happened. Read about Washington Crick and Mark. It's something. It's like soap opera. The pettiness went back and forth between them. And I guess scientists, for all their intelligence, like children sometimes. But these names you see here, if you ever back and actually read what's happening, you'd be going, I didn't know this. I didn't know they didn't like each other. I didn't know they fought like that, cats and dogs. I thought they worked together. Nuh-uh. Nuh-uh. All right. Now, I'm going to stop here on 15 because I'm about to get into another whole shooting match. But so far, we found out now how they figured out the nucleus wasn't making proteins because it's in the Beryllium experiment. We also found out that the electron scope, which I reckon Claude, you credit for showing Palade, was to open the doors to discovering the ER and the mitochondrion and the ribosome. She mentioned Peterson. What's that lady's first name? You remember?
Shoot. I have it.
We're discussing a woman named Peterson in the documentation is cited for discovering these ribosomes before these two guys ever did. Her friends say she was doing this like in 1930 something. And she asked me, why didn't she get her credit for it? I don't think I didn't tell you about it. Her friends knew, and her friends later on published. But when these two guys actually wrote the paper and put it in Nature magazine, that's called publishing. And they get the credit no matter how you know. White folks discover something and they'll mail letters to themselves.
Mary Locke Peterman.
Is it Mary? Okay. Some folks say, if you ever write a song, put those lyrics in an envelope, mail it back to yourself, and never open it. And if someone plays and you play your song, and they record that song that you wrote, and their name and they get the money for it, you go, I want to sue you. You can't prove. Yeah, I can. I got the address myself in 1950 where I wrote this song, and the court will open it, and I'll prove that's my song, and you owe me money. That's better than copyright. Write your song, seal it in a letter, put it in a deposit box, hang on to it. But that's your proof. That is your song. Just because you copyright it, because I can hear you sing a song and I can write the words down. And because I am Travis Tritt or some famous writer, they won't take me more seriously. And I get all the money for a song that Bobby Ted wrote. But Bobby Ted can't prove it. But I forgot, Bobby Ted, said sad, they can't prove it too. All right. Let me show you my new scope.